Okay, good morning everybody. It's good to see you after the holidays. As you know, you're nearing towards the end of the current course you're having in the module. Do you know what course you're in now? Okay, yeah, genetic information and its expression. And the course is going to end in a couple of days, I think. So in today's lecture, what I'm going to try to do with you is basically take what you've learned in genetics and molecular biology and biochemistry and show you how this knowledge can be used, or how you're going to use it once after 5.5 years, you're going to be doctors and you're going to be treating your patients. So how you can use this knowledge to treat your patients. So today's lecture is going to be, the title of the lecture is Principles of Anti-Cancer Therapy. We're going to be talking about how we can treat cancer in patients using what we know about genetics and molecular biology. Before we start talking about the actual principles of treatment, or the actual ways how to do that, I'd like us together basically to compile a list of differences between cancer cells and normal cells. Because the purpose of cancer treatment is obviously to get rid of cancer, of basically kill or remove the cancer cells. And in order to be able to do that, we have to be able to pinpoint some differences between normal cells and cancer cells. Because if we don't do that, then if we use any substances that kill all the cells, we obviously are going to kill the patient. Make sense? You seemed a bit passive today. So, so the idea is, in order to be able to treat cancer, we need to be able to see the differences between normal tissues and cancer tissues. So I'm going to give you 2 or 3 minutes. Just talk to each other as you sit around, by basically your friends around you, and try to come up with as many differences between cancer cells and normal cells as you can. Okay? So let's have 2 or 3 minutes now. All right. I think 2 or 3 minutes passed now. So just shout at me what you came up with.
Immortal. Immortal.
Immortal. Okay. By that, presumably you don't mean that there's no way to kill them. But what you mean is they normally don't die, they just keep replicating. Okay?
Proliferation without stop.
Okay, which is quite close to immortal. But I'll put in high proliferation or something like that.
They're almost undifferentiated.
Okay. Okay, less differentiated.
What about metastasis?
Okay, so they're able to metastasize. Okay. Let's put able to spread.
Lack of contact inhibition.
Say again?
Lack of contact inhibition.
Okay.
Resistance to apoptosis.
Okay.
They can accumulate in a very high concentration, although the volume is limited.
Say again?
They can accumulate or divide up to a very high concentration, although the volume is limited. Not concentration, loads of cells in a very little room, even though usually it couldn't be.
Okay.
I don't know how to put it like.
Because we're looking for differences between normal cells and cancer cells. So normal cells cannot proliferate, cannot grow into compacts.
Yes.
So what's the difference? I mean, I think you're right, but try to put it in a different way.
It's cell adhesion, isn't it?
What about it?
I'm not really sure.
There is no cell adhesion between them. But this comes very close to this contact inhibition thing.
Yeah.
There is actually cell-to-cell adhesion, because you do get solid tumors and the cells do adhere to each other. So what's the difference between, let's say, normal liver and a tumor? You think there are more cells on a smaller volume, in a cancer? Okay, okay, okay. Let's call it abnormal growth. Okay, is it too vague? So tell me and I'll write it in the
I'll
report.
think about it and then
Okay.
Normal cells, they will stop dividing if they don't have more room. But tumor cells will not stop dividing.
Okay, which probably goes under no contact inhibition, doesn't it? Yeah. Is that what you meant? Okay, okay. So it's a lack of contact inhibition, which normal cells do have. Okay.
More active telomerase.
In some tumors, but yeah, okay.
This belongs to immortal as well, doesn't it?
It goes to?
To immortal? Kind of immortal.
Sort of. Yeah.
In a tumor cell in a female individual, all the tumor cells will have the same pattern of X activation.
Okay, so that means that they grow.
One cell origin.
Okay, so can I put it as clonal growth?
Yeah.
Okay. Because it's not just about X inactivation, it's basically all the genome is pretty much.
It can be invasive.
Okay.
Chromosomal instabilities.
Excellent. Chromosomal instability. Why do cancer cells have chromosomal instability? Okay, so excellent. So most cancer cells have mutations in the repair systems, in the DNA repair systems, and that's why their DNA, their genome is more likely to be damaged. Yeah, that's absolutely correct. There's one other reason that sort of has to do with telomeres and telomerase. Okay, actually most tumors do not have telomerase, or many tumors do not have telomerase activity. So what happens is, as they keep dividing, their telomeres are getting shorter and shorter and shorter, as you all know the mechanism. And once, basically, after the replication starts eating into the chromosome itself, into the coding DNA, the chromosomes, the damaged chromosomes, starts basically sticking to each other and breaking, etc. So one of the major reasons why there's chromosomal instability in cancer cells is that the telomeres are getting shorter and shorter. The cells don't die. Normal cells would die. When the telomeres shorten too much, they die. These cells don't die. They just keep dividing. But the telomeres, or their chromosomes, are getting shorter and shorter, which means that they get damaged very easily. Does it make sense?
Yeah, but is there an end to this? Like if the chromosome is used up at some point?
Oh yeah, then the cell just dies. But there are so many other cells, you know. But yeah, there is obviously a point where the actual DNA molecule gets so short that the cell is unable to survive. But obviously the shortening is different in different cells, and sometimes there are not very important genes at the end, so the cell can go on. Yeah. So sometimes the patient dies before the tumor dies, if you know what I mean. Anything else?
Cancer cells are able to form their own blood supply.
Excellent. So we call it neoangiogenesis. Okay, neoangiogenesis. So the tumor itself can form new blood supply so that it can provide for its substrate needs. Very important thing.
Doesn't the tumor cell use more energy than normal cells? No?
Well, sort of. So the suggestion was that tumor cells use more energy than normal cells, that they have higher metabolism. That's sort of true. It depends which cells you compare it to. Because if you compare it to brain cell, to working brain cells or working cardiac myocytes, it's probably not true. Compared to normal stationary tissues, it's definitely true. But well, working muscle tissue is actually very high. So it's a bit tricky to compare and say that it's higher or lower. But there are definitely metabolic differences. So I'll put in metabolic differences. I'll explain what they are. Since about 1930s or something like that, it's been known that cancer cells have most cancer cells have very strange metabolic, basically they use some weird metabolic pathways to get their energy. The phenomenon is called Varberg effect. Obviously called after the guy who discovered it. And what Varberg discovered was that most cancer cells use almost exclusively glycolytic pathway to get their energy. So they don't generally rely on mitochondrial oxidative metabolism. They just use glycolysis and that's that. Nobody knows why that is the case, but it is a very striking difference between normal cells. I mean, obviously we do have normal cells that use exclusively glycolysis. What are they? Red blood cells, some other ones, okay? But there are obviously tissues in our body that use glycolysis either all the time or most of the time. But cancer cells seem to be very strange that if you have liver cancer, normal liver will use oxidative metabolism while the tumor will use mostly glycolysis. Strange, nobody knows why that is the case, but it's true for many, many tumors. Okay, so Varberg effect, the metabolic differences. What else?
Can viruses invade cancer cells?
Can
Viruses invade?
sure, yeah, they can. Or were you trying to say they can't? Or
I'm just asking if they can.
yes, I mean, yeah, cancer cells can be infected with viruses, yes.
The disease can progress. So the disease can happen if virus infects tumors, so you have two diseases at once.
I'm not sure where you're heading with that.
Don't worry.
So yeah, viruses can infect tumors, but they can infect normal cells as well.
Don't worry.
You had an idea, so try it.
that's fine.
No? Okay. All right. What else? What other differences can we find? Yep.
Maybe an activation of tumor suppressor genes.
Sure. Yeah. Inactive tumor suppressors, yeah. At least some of them. And on one side we have tumor suppressor genes, on the other hand we have, okay, activation of oncogenes. Now when we say activation of oncogenes, does it mean that normal cells have inactive oncogenes? Yeah, okay, so there's a tricky nomenclature thing, whether we call the normal genes proto-oncogenes and the abnormal genes oncogenes. Very often you hear people calling all the genes oncogenes, and there's just abnormal activation of oncogenes in cancer. But you're correct, okay, let's leave it as proto-oncogenes and oncogenes, okay? So normal cells have proto-oncogenes, which obviously are active and working, because normal cells do require proto-oncogenes, and cancer cells have oncogenes. Either the same ones, more upregulated, or they're mutated and they're constitutively active. Anything else?
Could you explain the less differentiated, why are they less differentiated?
That's just an empirical fact. Well, that's difficult to say. They have no function, because some tumors actually are functional. For example, they produce hormones, even though they shouldn't, but they are kind of functional. So it's kind of hard to say whether they're functional or not. But generally what you see in tumors is that they have less differentiated characteristics than normal cells. So they are, let's say, a bit more structurally, they're a bit more boring than normal cells. You can't quite say, is that an epithelium or is it more like, you know what I mean? So like, let's say a tumor, to make it easy, a tumor derived from skeletal muscle, which actually is very, very rare, but they do exist, will not have the structure of normal muscle. Okay, it's going to look like more like a stem cell. Okay, so it's less differentiated. It's not always true. You get tumors that are highly differentiated, and usually the more differentiated tumor is, the less dangerous it is, and less let's say less aggressive. Okay, the more differentiated tumors are less aggressive. The less differentiated tumors are much more aggressive. They grow faster. They're much more able to spread. So what you see generally is that tumor cells are less differentiated than normal cells. But there are differences between different tumors as well. Some of them are more differentiated, some of them are less differentiated. Does everybody understand the word differentiated, what it actually means, what it stands for in histology? Yeah? Differentiated, undifferentiated? Not really? No? Okay. So let's imagine in an epithelium. You all know what an epithelium is, okay? So you have, let's say in the skin, have you done the structure of the skin? Okay, so at the bottom you have the basal cells, basically stem cells for skin. They are kind of boring cells. You can't say, well, they look, you just look at a basal cell, you know exactly what it looks like, okay? Unlike the more differentiated cells where you can see a typical shape, a typical structure, typical proteins that are expressed, etc. For the less differentiated cells, for the stem cell, you can't say that. They just look kind of, ordinary. They just look like, there's nothing specific on them. Okay, so that means they're less differentiated. But as they differentiate more and more, so for example, stem cells for the skin and stem cells for the brain, let's say, would probably look very similar to each other. But their differentiated versions, so keratinocyte and a nerve cell in the brain, will look completely different. Okay, a nerve cell has axon and dendrites and all sorts of things, while keratinocyte, has a very specific shape, etc., and produces keratin, etc., etc. Okay, so even though the less differentiated cells may look alike, their differentiated version have very striking, very specific structure and shape. Okay, so that's what we're talking about. Cancer cells generally tend to lose the differentiated characteristics. They tend to look alike, not completely alike. You can still distinguish them, but. Okay, so that's about differentiation. Any other suggestions? Yeah.
They attack surrounding tissue.
Okay, well we have it under they're able to spread or invade. Yeah.
They need a host. They need a host.
They need a host. Okay, what do you mean by that?
Like viruses. They need.
So cancer cells need a host in what way? I mean, you're right. You're absolutely right. I mean, you don't, walk on a street and in a puddle you find cancer. It doesn't work like that. It has to grow. I mean, cancer grows inside our bodies. But then again, cancer is our tissue. Okay, so you're right. It has to grow in a host because it cannot live. You can't take cancer out of the body and just have it grow. Well, you can, but anyway, that's not the normal way to do it. Okay. So yeah, you're right. They do need a host, but they're basically part of our body anyway. Okay? I think we actually covered most of the things that I wanted to mention. I'll just once again stress, when we talk about chromosomal instability, we talked about impaired repair mechanisms. That's very important. Okay? Cancer cells generally have impaired repair of their DNA. They can't repair the DNA when it's damaged as well as normal cells. And that's actually very important, as we'll see in a minute. That's very important for cancer therapy. Okay? So impaired DNA repair, it's very important. And the other two things that I'll mention, we sort of mentioned them as well, but I just kind of want to reframe them slightly. The inactivity of tumor suppressor genes and the activation of oncogenes. You all know that that's the necessary step for cancer to develop. But what's very important for anti-cancer therapy is that, let's say that normal cells have active proto-oncogenes, or which generally are the same genes as oncogenes. Cancer cells seem to depend on those oncogenes much more than normal cells. Because normal cells don't need to now I'm generalizing, okay? There are different dependence between different tissues. What I'm trying to say is normal cells, if you block their activity of their proto-oncogenes, will probably survive. Okay, it may not be very happy, but it will probably survive. Cancer cells generally are extremely dependent on these superactivated oncogenes. If you block them, it's very likely that the cancer cell will die. Why do you think that's the case? Why do you think cancer cells are so much more sensitive to inhibition of their oncogenes? Yeah? They can't repair them. Well, they're generally proteins, so you don't really repair proteins. I mean, okay, we're talking about oncogenes. I'm talking about the proteins, the products of oncogenes. But yeah, okay, they are genes, but I'm talking about the proteins. Because they're dependent on the proteins, not so much on the genes. They need the proteins to keep them working. Can anybody come up with an idea why cancer cells are so much more needy? They need the oncogenes to work a lot more than normal cells. Yeah?
Because oncogenes lead cancer cells to spread. Maybe.
Oh, you mean that the oncogenes are basically, okay.
The reason of...
Yeah, so it's the idea of a selfish gene, that basically the oncogene is trying to spread. Okay, yeah, interesting.
If there's a cancer when the oncogene is very active, and the tumor suppressor gene is still active, but not that much anymore. So if the oncogene is gone, the tumor suppressor gene will get upper hand. Okay,
that's practically the answer. In cancer cells, in normal cells, there is usually a very nice balance between, let's say, tumor suppressors, suppressors, and oncogenes. In a cancer cell, there is no balance. Cancer cells generally are very ill cells. Okay, they have damaged chromosomes. They produce weird proteins. They have weird metabolism. They are not well. Okay, those cells, and they only can survive if they have overactivated oncogenes and basically protective genes that can allow them, even with all these damages, to go on and survive and proliferate. So they are much more dependent on this support from oncogenes than normal cells. Because normal cells are generally not damaged, okay? They're fine. So if you decrease the activity of kind of a supporting oncogene in a normal cell, it's going to survive. In a cancer cell, that's usually not the case. And that's what we use in cancer therapy, and we'll talk about it in just a second. Okay, any questions about the differences between normal cells and cancer cells? If not, let's get to the next stage and let's talk about the actual anti-cancer therapy. So what are the basic modalities, what are the basic ways how to treat cancer? Yeah?
I have a question.
Yeah?
Do they necessarily have inactive tumor suppressors all the time?
So the question is, do cancer cells have inactive tumor suppressor genes all the time? No. Some of them are active, some of them are inactive. Depends which cells, depends which tumor. Sometimes, I mean, very often you have to have at least some tumor cells inactive. Some tumor suppressors inactive, because otherwise the damaged cells would just apoptose basically without the tumor suppressor. I mean, with the tumor suppressors present. So usually there are at least some tumor suppressor genes that are repressed or mutated or something in cancer cells. But that doesn't mean that all of them are in all cancer cells. Okay, so it depends. It varies.
So it's not enough if they're just suppressed, but they have to be inactive. So if
no, they can be just suppressed. Okay. Okay, so what are the basic modalities, what are the basic ways how we can treat cancer? That's a question. Okay, so one... Okay, so first way is to cut it out. You said that that's used for benign tumors, but actually it's used for malignant tumors as well. But you're right that when there are metastases, it's not a curative way, okay? So if we have tumor that has already spread into the body, we can still cut it out, and very often that's what's done. But that doesn't cure the patient. If the tumor, whether it's benign or malignant, and it's still localized, then you can cut it out and you'll basically cure the patient. Okay? So first modality, and it's by far the most used modality, well, it's very often used, is surgical removal of the tumor. Okay? Obviously, that only works for solid tumors. Do you know what I mean when I say solid tumors? What's the opposite of a solid tumor?
Leukemia?
Yeah, so usually it's blood-derived tumors like leukemia or lymphomas, okay? Because there we don't have anything solid, but they are quite literally, leukemia is quite literally a liquid tumor in a way, okay? So solid tumors are the tumors that actually have solid structure. So obviously surgery is used for solid tumors. You can't excise leukemia, as you probably understand. Okay, surgery is one. We're obviously not going to talk about it today because it's not very interesting from a cellular point of view. There are other modalities though. Yeah?
Radiotherapy.
Radiotherapy, okay, radiation. How does radiation work to kill, or what kind of radiation do we use?
Gamma, for example.
Okay, so we use gamma. What else? It's gamma rays. What else can we use to treat cancer? Okay, ultraviolet, UV light. Sort of. Let's leave UV light aside for a minute.
Don't you use X-ray?
You can use X-rays. Okay, you can use gamma rays, X-rays.
Inhibition of inhibitors or of the proliferation. That's more.
Okay, so you're talking about other modalities, so about chemotherapy. Okay, let's leave chemotherapy as the last one because that's going to be the bulk of the lecture, basically. Okay, so let's finish off the radiotherapy. So we have gamma rays, we have X-rays. Are there any other? On the radiotherapy? Okay, have you heard about neutrons or heavy ions? Okay, so these are quite new modalities, well, relatively new modalities of treatment where you don't use electromagnetic waves such as gamma rays or X-rays, but you use actually accelerated either neutrons or accelerated heavy ions that are basically pushed into the tumor and damage the tumor. Okay, that's radiotherapy. Again, we're not going to talk about it very much now. How does, let's just go back to UV light and visible light. Why did I kind of hesitate with UV light or visible light or infrared light? Why do these wavelengths of electromagnetic radiation are not as effective in treatment, or maybe completely ineffective in treatment of cancer? Why can we use gamma and X-rays but not UV? They have more energy. And what do we need the energy for? So basically my question is, what do gamma rays and X-rays do to the tumor?
Ionizing?
Excellent. They're ionizing. So X-rays and gamma rays are ionizing radiation, while UV and larger wavelengths are not ionizing. Okay? So what we do in radiotherapy, we're trying to ionize, in a way, the tumor, and that kills them. The actual ionization, even though it's not quite ionization, that happens in the tumor when it's irradiated by gamma rays or X-rays, is the photolysis of water. So water molecules are split into hydrogen radical and a hydroxyl radical. And hydroxyl radical is extremely reactive and will react with everything in the cell and will just destroy the cell. Okay? Especially hydroxyl radical is very happy to react with DNA and completely disrupts the DNA. So that's what we do by irradiating a tumor with gamma rays or X-rays. Okay? Photolysis of water. Photolysis. Okay? Photolysis of water. You don't have to know that, but maybe you should know it from biophysics rather than here. But anyway. Going back to UV light and visible light, you can use even that to destroy tissues. But on its own, UV light and especially visible light is unable to damage the tissue directly. Okay? I mean, you all know that UV light can do it when you use a very short wavelength. But what you can do, and what are actually some of the newer modalities of treating tumors, is that you can use a drug called a photosensitizer, and then use visible light, for example, and shine on the cells containing the photosensitizer, and that kills them. So there are chemicals that basically make cells more sensitive to normal radiation, and we can use that to kill the cells. Okay? But that's all about radiotherapy. And let's go back to the final modality, and that's the one we're interested in today, and that's chemotherapy. Okay? So again, we said that there are a number of differences between cancer cells and normal cells, and we use those differences to kill specifically, or at least more specifically, cancer cells and not normal cells. Obviously, since we said that cancer cells are our own cells, it's much more difficult to kill cancer cells that have just slight differences from our cells than it is, for example, to kill bacteria. When we have bacteria in our body, bacterial cell is so much different from our own cells that it's actually fairly easy to kill just bacteria and not the body. In cancer, that's a lot more difficult because the cancer cells and normal cells are actually very, very similar, even though we talked about many differences. So let's now talk about the groups of drugs that are used in chemotherapy, and with each of them I'll explain what they do and how they do it. The first group I want to talk about is a group of drugs called alkylating agents. What does it mean, alkylating? No idea.
Something with the alkanes.
Something that alkylates, yes, that's true. What does it mean to alkylate? What is alkyl? Or alkyl? Okay, yeah, just speak up.
You just add them, I guess, to
Exactly, so these are drugs that basically take a hydrocarbon residue or something like that and attach it to something. That's what they do. Okay, so they're usually very reactive compounds that just bind to various cellular structures. And the most important structure is DNA. So what alkylating agents do, they alkylate, so they bind part of their molecule to DNA. And obviously that damages the DNA, impairs transcription, translation, replication, etc., etc. So this is what alkylating agents do. I will draw one of them, the structure, because it's fairly, you don't have to obviously draw it. So this is one of the agents used as an alkylating agent, one of the drugs, one of the compounds. And the alkyls, the residues of hydrocarbons that are used, that bind to DNA, are basically these bits. So the chlorines go away, and you get two very reactive bits that can bind mostly to DNA. And obviously, if you have two bits, that means that one of them usually binds to one nucleotide, the other one to another nucleotide, and that binds the double helix of the DNA together by covalent bonds. And basically, it's impossible to unwind it. Okay, does it make sense? What alkylating agents do? Yes?
Yeah, but where's that molecule come from? Like,
It's a drug, so you give it to a patient. Okay? It's a synthetic drug. Do you all understand how alkylating agents work in principle?
Is it precise for cancer cells, or is it?
Well, okay, whether it's specific for cancer cells, it isn't. It will alkylate DNA in all the tissues in the body. But what makes it a bit more specific is what we talked about, that cancer cells are very ill. So any damage that we do to cancer cells usually damages them more than normal cells. So when you have a normal cell that has several of these molecules bound to its DNA, it doesn't usually damage it too much because the repair systems are working, so this thing is excised. The cell doesn't usually divide very much, so it doesn't really cause a huge problem if you have alkylated DNA. In a cancer cell, because the repair mechanisms are not functioning and there are all sorts of other things, the cancer cells will suffer much more from this alkylation than normal cells. Okay? So this agent specifically, this agent is not specifically, it doesn't work specifically in cancer cells. It works everywhere in the body. But normal cells are less sensitive to it. Okay, make sense? Good. There was a question there? No? Yeah?
Yeah, I was just wondering, is it nitrogen?
That's nitrogen. That's nitrogen, yeah. But I said don't draw the structure. It's, I mean, it might be interesting, but you don't have to know it, obviously. What is kind of interesting is the history of these alkylating agents. Because before they were developed as drugs, they were developed for an entirely different purpose. I mean, you can probably imagine that these types of compounds that are very reactive and will bind to all sorts of things in the cell are going to be very toxic. Do you see that? Yeah, it's very likely, isn't it? And actually, the first use for these drugs in the early 20th century was in the war. Basically, they were used as toxic gases to kill people. Well, not so much to kill people actually, because they're not that toxic, but to incapacitate them. Because what the exposure to these kind of compounds does to a person, I mean, when they are exposed to the gas, not to a cytostatic agent that's used to treat cancer, they basically have blisters on their skin. So the contact with the skin causes huge blisters, basically burns, chemical burns. So it doesn't necessarily kill the soldier, but it incapacitates them. They're unable basically to fight for a very long time. The original compound, and that's the reason why I drew this drug that's used. The original compound used for killing people looked like this, and it was called mustard gas. I don't know if anyone's heard of that. Okay, so this is the original First World War, one of the most dangerous gases that was used to kill people, apart from chlorine. This was mustard gas. Okay, very dangerous, very incapacitating. And as you can see, what later on people did was they just slightly modified it, and it was used to treat cancer. Yeah?
How long does it take until it binds to the DNA?
You mean with mustard gas, or with this thing? I mean, this thing can be given intravenously, and obviously it happens relatively quickly. Once it gets into the cell, it will bind to something. Okay? With mustard gas, after the exposure, it usually took about 24 hours before the symptoms develop, because obviously it just binds to DNA. So until the cells need to do something with the DNA, pretty much nothing happens. Okay? The interesting thing in the history is that, I mean, why people started thinking about using this to treat cancer was that people exposed to mustard gas usually suffered from very low white blood cell count. Okay? So it damaged their bone marrow, and they had much less white blood cells. So early in the 20th century, people were starting, well, let's say in the 1930s and 40s, they started thinking, okay, well, if it decreases the white blood cell count, we could use it to treat leukemia. Because in leukemia, you have huge numbers of white blood cells. Okay? Not normal white blood cells, but tumors. So they tried, basically, they made different derivatives and gave them to people with leukemia and lymphomas, and they found out that they're actually working quite well. So later on, these alkylating agents were used for other kinds of tumors, and they're still used to this day. They're one of the most important groups of anti-cancer drugs, alkylating agents. There are many, many more with much more complicated structures, but the principle is the same. Okay? Now, I'll show you another alkylating agent that doesn't actually alkylate, but it's usually put in the same group. And once again, I'll draw the structure, because it's very, very simple. This drug is called Cisplatin. Sometimes you see it without the hyphen. It's called Cisplatin. Do you think anybody could name the compound? What kind of compound is it? It's not really an amine, no. It's actually a complex. It's not a normal covalent compound, it's a complex. It's a coordination compound with platinum as the central atom, okay? And 2 molecules of ammonia and 2 chloride ions bound to it. Okay? It's a complex of platinum. Okay? You all look very confused or something. Have you heard of coordination compounds, complexes? Never ever. Some of you have. Hmm? Exactly, yeah. So these 4 things are ligands that bind to platinum. Okay? 2 chloride ions to 2 molecules of ammonia. Anyway, so it's a complex, it's a coordination compound, and it's called cis because in fact it is the cis isomer. Okay, cis-trans, you should definitely know that. Okay, how would transplatin look like? Hmm? Okay, so the 2 ammonias would be here, and the 2 chlorides would be here, so they would be basically opposite each other, okay? So you can get actually isomers even in complexes and coordination compounds. But anyway, cisplatin is usually put into the group of alkylating agents, even though, obviously, it cannot really alkylate, can it? I mean, there's no alkyl there, there's no hydrocarbon. But what it does, it actually goes into the DNA and binds the DNA into a complex. So basically these ligands, at least some of them, are removed, and the nucleotides, the bases of the DNA, serve as these ligands. So the platinum goes into the DNA and binds the surrounding nucleotides and binds them very, very strongly. So in effect, it works in a very similar way as the alkylating agent that basically binds to one and the other one and just keeps them together. The platinum does exactly the same thing, even though it doesn't use covalent bonds, it uses coordination bonds, but that's really a tiny difference. Okay, so cisplatin, a very, very, very important anti-cancer drug used in many cancers, usually solid tumors, but very important. Okay, any questions? Okay, if not, let's have a 5-minute break. So let's start with the first molecule. So what is it and what could it do to a cell? And why?
Resembles purine. Purines, adenosine.
Okay, it's a purine, definitely. And which purine specifically does it resemble?
Maybe both.
Yes, it does resemble both because it's a purine, but one of them a bit more than the other.
Guanine has the O instead of the sulfide, right?
Excellent. Okay, so which one was it?
Guanine.
Guanine. Okay. And you correctly said that instead of sulfur, guanine has oxygen. Okay, so in guanine this would be oxygen, but here it's sulfur, so it's called thioguanine. Okay, thioguanine, specifically 6-thioguanine. Right, so it resembles guanine, but it's not quite. So what could it do if it's given to a cell? What will happen to it? Okay, so we'll replace guanine. Where? In the DNA. Okay, so it gets incorporated into the DNA, and what happens then? Why will it not be able to form double helix?
Because it's made by hydrogen.
Okay, there's something about hydrogen bonds. Okay, the way, okay.
Impaired complementary base pairing.
Excellent, so you're both right. What will happen is that this thioguanine will not base pair normally. It will not base pair the same way as guanine would, okay? The actual story is a little bit more complicated because it becomes methylated, etc., etc. But the basic problem is that thioguanine will not behave as guanine in the DNA and will impair replication and all sorts of other things. Okay? So 6-thioguanine, a very important, well, an important anti-cancer drug, And it works by being incorporated into the DNA and then impairing the normal metabolism of DNA, like replication. Yes?
Does it have a higher affinity to be bound into the DNA than the normal guanine, or why is it?
I don't know if it has a higher affinity. I think it probably has the same affinity as normal guanine. I imagine maybe even a lower affinity. But the thing is, if you incorporate only few thioguanines instead of normal guanines, you basically damage the stretch of DNA quite badly. Okay? So I don't know what the actual affinity is. I think it's probably even lower than normal guanine. But it only takes a few thioguanines incorporated to damage the DNA. Okay? And once again, it's not specific for cancer cells. Okay? Normal cells will obviously incorporate into their DNA as well. But as we said before, cancer cells tend to be much more sensitive to this kind of DNA damage than normal cells. Okay? That's why we use it to treat cancer. Good. What about the second compound? Actually, let's do the third one, and let's leave the middle one for as the last. So what about this one? What does it look like? Yeah, like a nucleoside. Which one?
Is it maybe cytosine or cytidine?
Okay, cytidine. Okay. Okay, it looks like cytidine. Can everyone see it, at least sort of? Now, what's the difference between this thing and cytidine? It's quite subtle, but it's enough to do some damage.
The end group is changed with O.
Which one?
I thought it was maybe 2 O groups in cytidine.
This one?
Yes.
No, if this was an O group, it would be a different base.
Yeah.
Okay? I'm not going to say which one, but this is definitely cytosine. There's nothing wrong with this. Okay? This is cytosine. But why is it not cytidine?
Because it's not a deoxyribose, but a normal ribose.
Okay, it's not a deoxyribose. So what is it? It's not even ribose, actually. That's the problem. It's not fructose. It's a different sugar, and you obviously won't know it. It's called Arabinose. Which is not that important that it's Arabinose. But the difference between Arabinose and ribose is that this OH in ribose is here. That's the only difference. Okay? So this OH group has just the opposite configuration. Okay? And that makes it stop being ribose and becomes Arabinose. Now, what happens when you give... So the whole thing is called cytarabine. Okay, to say that it has cytosine and Arabinose, cytarabine. What is it going to do to a cell when it encounters this drug? Which process is likely to be impaired by this? Well, it's going to get incorporated into well, mostly RNA actually, but DNA potentially as well. Okay? But then, once it gets incorporated, because the configuration at this end of the sugar, because you'll recall that DNA and RNA basically go, okay, like that, as a chain. Here, because the configuration is different, the polymerization of RNA or DNA containing this modified nucleotide will stop. Because the DNA polymerase or RNA polymerase will be unable to continue the chain because this sugar is not ribose or deoxyribose. Okay? So this drug primarily damages DNA polymerization and RNA polymerization. Okay?
What's the name? Cytarabine?
Cytarabine, yes. Cytarabine. I-N-E. Right, what about the middle one?
Fluorothymine.
Good thinking, but not quite. It's fluoro something definitely, but not thymine.
Not thymine, uracil.
It's fluorouracil. It's 5-fluorouracil. It's very good thinking. So what's the difference between uracil or 5-fluorouracil and thymine?
There's this fluoro.
Okay, so obviously the fluor is not there. The fluorine is not there.
The CH3 group.
Okay, and there's a methyl group instead. Okay, that would be thymine. What will 5-fluorouracil do to a cell? What will happen to it?
Same as with the first one.
So it will be incorporated into what?
The polymerase can't work or stuff
Why can polymerase not work?
because it's not the same as the normal pyrimidine. It's a fluorine instead of a methyl group or nothing.
Okay, yeah, that's one possibility. One possibility is that it resembles a pyrimidine, a normal pyrimidine base, but it doesn't quite fit all the requirements for a normal pyrimidine. Okay, again, very good thinking, but in this case, that's actually not what happens. Okay, because fluorine is actually not that much different from hydrogen to some extent. So, in fact, the problem here isn't that 5-fluorouracil gets incorporated into nucleic acids. It will get incorporated to some extent. But there is another problem, and it has to do with the resemblance of this with uracil and sort of with thymine. How is thymine synthesized? Does anyone remember how thymine is made? What is it made from? What is thymine made from? Well, we just said that the difference between uracil and thymine is just a methyl group. Is that right? Okay. This is uracil, and if this was CH3, it would be thymine. So thymine is made by methylation of uracil. Okay, that's how it's made. Now, what if this enzyme called thymine synthetase will take 5-fluorouracil instead of uracil, and we'll try to methylate it? Is it going to be possible? What do you think? Is it going to be possible? Okay, it's not going to be possible because there's already fluorine there, where it should put in the methyl group. Okay, so what 5-fluorouracil, the main activity of 5-fluorouracil is blocking thymidine synthesis. Now, there's a curious thing about cells, and that's that when they don't have enough thymine or thymidine, they die. It's very strange because for other bases, they're much less sensitive. But thymine seems to be a very important base. So if a cell doesn't have enough thymine, it will basically apoptose. And that's what we use when we treat some cancers with 5-fluorouracil. Okay? You give it to the cells, they can't make enough thymine, and they apoptose. Okay? Once again, the idea is that the cancer cells will be more sensitive to this than normal cells. But obviously normal cells will suffer from it as well. Okay? 5-fluorouracil. This group of compounds, this group of drugs, are called anti-metabolites. Obviously because they inhibit some aspect of metabolism of nucleic acids. Okay, of nucleotides or nucleic acids themselves. The last member of the group of antimetabolites is a very different compound that doesn't look like any of the bases. So I'm not going to draw it because it's a bit more complicated structure. But I'll definitely mention it by name because it's, again, one of the most important anti-cancer drugs used. And it's called methotrexate. Now, methotrexate is not an analog of a base of a nucleotide or nucleoside or pyrimidine or purine. Okay, the structure is completely different. But it also affects metabolism of nucleotides. Methotrexate is the inhibitor of an enzyme called dihydrofolate reductase. Do you want me to write down the name? Dihydrofolate reductase. Okay? Dihydrofolate reductase. What does dihydrofolate reductase do? Should be quite easy to guess from the name. But what does it reduce?
Dihydrofolate.
Okay, it reduces dihydrofolate into into tetrahydrofolate. Okay? Now tetrahydrofolate is needed for one step in, well actually for two different steps in the synthesis of nucleotides, of bases. Does anyone know where we need folic acid in the synthesis of... Which steps in the synthesis of, or which bits of the synthesis of nucleotides require folic acid or tetrahydrofolate or its derivatives? Okay, there's one step in the synthesis of purines which requires tetrahydrofolate. And the other step is the synthesis of thymine, the synthesis of thymidine. The methylation of uracil into thymine requires tetrahydrofolate. So by inhibiting the dihydrofolate reductase using methotrexate, we decrease the levels of tetrahydrofolate in the cancer cell, well, in fact in all cells. And that leads to decreased production of thymine, and we already talked about what that causes. Okay? Does it make sense what methotrexate does? With methotrexate, there is a very interesting way to basically leverage a difference between another difference between normal cells and cancer cells. Because cancer cells, at least some cancer cells, are unable to transport enough folic acid from the blood into the cell. Okay, because they're due to various mutations, etc., some cancer cells can't really get enough folic acid from the blood. Okay? So what is very often done with methotrexate is that a patient is given basically a lethal dose of methotrexate. Okay? A huge dose of methotrexate. But shortly after that, he or she is given an antidote to methotrexate, which is basically folic acid. Okay, it's actually called leucovorin because it's a derivative of fatty acid. Of folic acid. Leucovorin. It's an antidote to methotrexate. But because cancer cells are defective in getting folic acid or its derivatives into them, they will suffer by the inhibition caused by methotrexate much more than normal cells that will be damaged by methotrexate, but leucovorin will save them. Okay? So methotrexate gets everywhere. Leucovorin only gets into normal cells. So normal cells will be fine, or at least sort of fine, while the cancer cells are supposed to die because of the inhibition caused by methotrexate. Does it make sense? Yeah? Methotrexate and leucovorin. Good. Any questions about anti-metabolites? No. Okay. Now we're getting from drugs that we talked quite a bit about and about the mechanism of effects, and we're going to get into groups of drugs that I'm just going to mention and talk very, very shortly about. The next group of anti-cancer drugs that I want to talk about inhibit the manipulation with DNA. So they don't inhibit the synthesis necessarily, but various manipulations with the DNA. It will become clearer when I start talking about them. One of the things that you've probably heard about that, but one of the things that is one of the problems with manipulating with DNA in our cells, or in bacterial cells for that matter, is that the DNA molecule is basically, it's a double helix and it's very, very long. So when we need to replicate it, for example, we have to unwind the double helix. But because the molecule is very, 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 very long, basically the unwinding causes tension in the rest of the molecule. Okay? You've heard that. Okay. And there are enzymes in our body that can basically relax the tension. Do you know what they're called? Excellent. Topoisomerases. Topoisomerases. Okay? Which basically means that they isomerize different topological structures. But yeah, anyway. Topoisomerases. So the group of drugs that I want to talk about now are basically inhibitors of topoisomerases. Okay? And you can imagine if you inhibit the ability of the cell to relax the tension in the DNA, it will inhibit manipulation with the DNA, it will inhibit replication, potentially transcription, etc. Do you understand how topoisomerases and their inhibitors work? Not quite? Okay. I can't really go into detail about topoisomerases, so read up on that at home. One group of topoisomerase inhibitors are called intercalating agents. Does anyone know what intercalating agents are? No? Intercalating agents are molecules that basically bind into the DNA in a similar way as cisplatin that we talked about. They don't bind to anything covalently. They just sit in the double helix and basically impair the ability of the double helix to unwind. One of the intercalating agents, one of the major groups of intercalating agents, is called doxorubicin. It's an antibiotic produced by Streptomyces. And it basically sits, it goes into the DNA and just sits there. But also the other activity, and that's why we talk about doxorubicin in the group of topoisomerase inhibitors, is that doxorubicin also inhibits topoisomerases. Specifically, doxorubicin inhibits topoisomerase 2, because there are two different kinds of topoisomerases, but that's not something that you need to remember. Of course, we also have inhibitors of topoisomerase 1. The two topoisomerases work differently. And one of the inhibitors is called topotecan, which is the inhibitor of topoisomerase 1. Okay? So this is super quick talk about topoisomerase inhibitors. The two that I mentioned are doxorubicin, a very, very important anti-cancer drug used for many, many tumors, and topotecan as an example of topoisomerase 1 inhibitor. Any questions about topoisomerase inhibitors?
Can you just explain it again what they do?
Sure. So topoisomerases are enzymes that release the tension in the DNA molecule. Okay? With the manipulation with DNA, and because it's a very long molecule, in bacteria it's a circular molecule, there are huge chromosomes. So whenever you do something with the DNA, whenever you unwind it, you create tension elsewhere in the molecule. And these enzymes serve to release this tension, because when the tension becomes too big, the molecule, basically any manipulations with the molecule are impossible, because helicases and all sorts of things cannot work with it. Okay? Does everybody understand what topoisomerases do? Good. Well, then these two drugs simply inhibit them. Okay, that's all you need to know. The other very important group of inhibitors of manipulation with DNA are microtubule inhibitors. So they are they are the inhibitors of microtubule, basically assembly or so microtubule inhibitors. Why are microtubules important for what do microtubules have to do with DNA?
And mitosis, they're required for the...
Exactly.
Mitotic
So most important, the mitotic spindle is made of microtubules. So if we damage microtubules, the cells are unable to divide. Okay? And that's basically the mode of action of microtubule inhibitors. Microtubule inhibitors come again in two groups, and both of them are derived from plants. Okay? So they are plant-based drugs. The first group are called vinca alkaloids. And they're derived from a plant called Vinca rosea. And I'll just mention one of them, which is vincristine. Just one example of the vinca alkaloids. The vinca alkaloids inhibit the polymerization of tubulin. Okay? So when vinca alkaloids get into the cell, they block the polymerization of tubulin. You can't form a mitotic spindle, and that's that. You can't divide. Okay? Make sense?
Doesn't that damage all the other cells as well?
As usual with all those drugs, yes, it will. But the cancer cells, the hope is that they will be more sensitive to this damage than normal cells.
What about other rapidly dividing cells?
They will be damaged by this.
So I guess you will...
Yeah, the side effects are typical for most of these drugs: bone marrow depression, anemia, alopecia, hair loss. Those are typical symptoms of that. Yeah, side effects. Yeah. The other group of microtubule inhibitors works in exactly the opposite way from vinca alkaloids. So vinca alkaloids block the polymerization. The other group, which are derived from taxus, okay. The most famous example is Taxol or paclitaxel. Taxol is the company name. Paclitaxel is the generic name. Oh, does anyone know what taxus is in English? It's yew. Yew tree. Okay? No? It's a coniferous, very kind of, yeah, it's slow growing. Hmm? Yeah, with little red berries.
Yeah, we heard about that yesterday.
Okay. So anyway, Taxol, paclitaxel works in the opposite way. So instead of inhibiting microtubule polymerization, it inhibits the depolymerization. So once the microtubule is there, the cell cannot destroy it again. And obviously in mitosis, you have to constantly build new microtubules and destroy them again. So with Taxol, with paclitaxel, this depolymerization is inhibited, and therefore cell division is inhibited. Once again, yes, it will affect normal cells as well. Okay, in the last 1 minute, I'll mention a very broad group of new treatments for cancer. And I call them, for my preparation, I call them specific treatments. All the drugs that we talked about now are not very specific. They will just affect everything in the body. But obviously, people have been trying for many, many years to develop drugs that would be more specific for the cancer cells and actually damage them more, or damage them specifically only, the cancer cells. The first members of this drug of specific treatment is fairly old, and those are hormonal treatments. Some cancers are very dependent on certain hormones, very often sex hormones. For example, breast cancer, some breast cancers are dependent on estrogens, and some prostate cancers are dependent on androgens, testosterone. So we can have drugs, and we do have drugs, that inhibit the action of estrogens, called anti-estrogens, and drugs that inhibit the action of androgens, anti-androgens. And they're both used to treat these hormone-dependent cancers. Not all of them are hormone-dependent, but when they are, these drugs can be very effective. These are fairly old. The newer drugs, the newer specific drugs, basically target very specifically certain signaling pathways or certain proteins, certain receptors that are only present in specific cancers or are upregulated in specific cancers. So for example, there are now drugs that can inhibit the neoangiogenesis that we talked about. So we said that some tumors, or many tumors, need to build their own blood vessels in order to keep their supply of nutrients and oxygen so that they can grow. Now there are drugs that can inhibit this growth of new blood cells, new blood vessels into the tumor. And obviously this will harm the tumor much more than normal cells, because normal cells in an adult, normal tissues in an adult don't really need to grow new blood vessels. Some of them do, but most of them don't. So inhibiting neoangiogenesis will hurt specifically the tumor, not normal cells. Okay? Two specific agents in this group, which I think you should hear about. One of them is retinoic acid. In one of the courses next semester, you'll hear that retinoic acid is a very important signaling molecule, especially during embryonic development. So retinoic acid is a signal that tells tissues whether they should grow, whether they should differentiate, etc. It's a very important natural signaling molecule in the body. In one type of leukemia called acute promyelocytic leukemia, don't have to know that, giving the patient retinoic acid causes these undifferentiated cancer cells, the leukemia cells, to differentiate. So they differentiate into pretty much normal white blood cells that which after that die because there are just too many of them. Okay? So these cancerous cells, undifferentiated cells that grow very rapidly, when they see retinoic acid in a certain dose, will differentiate into almost normal cells, which then die. Okay? A very specific treatment that doesn't really affect, in an adult, it doesn't really affect normal tissues. It does to some extent, but not hugely. In a fetus, in an embryo, it will be a very dangerous treatment indeed, because the embryo uses these signals to actually grow its fingers, etc., etc. So retinoic acid. The other one I want to mention is called imatinib, or the company name under which it's much better known, Gleevec, is a very specific drug. Gleevec, G-L-E-E-V-E-C. I-M-A-T-I-N-I-B. Yeah, not very nice word, is it? Imatinib. In the first lecture in this course, you heard about a disease called chronic myelogenous leukemia. Is that right? Do you all remember? Anyway, there is a drug, there is a disease called chronic myelogenous leukemia, and the interesting thing in this disease is that it has a very specific chromosome translocation called Philadelphia chromosome. Okay, the translocation basically causes this cancer because two unrelated genes get very closely together, and a fusion gene, a fusion protein is formed that basically makes the cells grow and be cancerous. Imatinib is a specific inhibitor of this fusion protein. So this weird protein that causes this cancer that's caused by this chromosome translocation can be specifically inhibited by this new drug. And as you can, and it's obvious that this drug will not really affect normal tissues because they don't have this weird fusion protein that's only present in cancerous drugs. So these specific drugs are, let's say, the future of cancer therapy. These drugs, all the other ones that we talked about, even though they're still very, very important in treatment, you can say that they are the past because they're not very specific. They just kill all the cells in the body, and we just hope that the cancer cells are more sensitive to them. These are few members of this new group of drugs that are much more specifically targeted to the cancer cells, and therefore they have much fewer side effects than the old ones. Okay, questions? Yes?
Is there any chemical cancer therapy that does not have a negative effect on embryology?
Oof, that's a tough question. If there's any treatment, any chemotherapeutic agent that does not, I don't know. I would have to really think about that.
No, but it's none of the ones we've had. Like all of them will affect.
Yes, all of them are teratogenic. Yes.
Actually, read something about that. It was in the news that a mother gave her life for her children because she didn't want to have the chemotherapy. So I guess there is no...
Most chemotherapy is contraindicated in pregnant women. As in, usually often the pregnant woman that has cancer, especially if it's acute cancer that needs to be treated, they usually opt for abortion. But of course, I'm sure there are cases where the opposite is true. Yeah, what is quite curious is that very often in pregnancy, some tumors are much more aggressive than in women that are not pregnant. That's probably the most likely reason for that is that in pregnancy, the hormone levels are much higher. And because some tumors are hormone dependent, they will just grow much, much faster. Just unfortunate. Other questions? Okay, if not, that's all.